Assalamualaikum warahmatullahi wabarakatuh. We are from Group 7. We're now presenting about the application of animal cell culture in cancer research. Okay, so the theme for our Group 7, we have Rabiatul Adawiyah binti Sulaiman, Nur Akilah Nadira, Amanina Diana binti Ahmad, Siti Aisyah binti Mat Yusof, Rozainah binti Muhammad Rizwan, and also Hayatun Nabilah binti Norjai.
The first effective development of cells taken from Mrs. Henrietta Lacks's cervical carcinoma was demonstrated in the early 1950s. Mrs. Henrietta Lacks's cells in culture revolutionized medical and biological research, enabling for several cellular, molecular, and therapeutic breakthroughs, including the development of the first efficient polio vaccine. This culture is now called HeLa. Animal cell culture is a technique for growing human, animal, or insect cells in a controlled environment. The cells might come from multicellular eukaryotes, pre-existing cell lines, or pre-existing cell strains. Animal cell culture became popular in the laboratory in the mid-1900s. However, the notion of keeping living cell lines apart from the original tissue source was found in the 19th century. In order to separate the cells from the tissues, disaggregation utilizing enzymatic or mechanical techniques can be used. The majority of cells in animal tissue culture are anchorage dependent and so required a solid or semi-solid support in the form of a substrate, which is referred to as adherent or monolayer culture. Others, on the other hand, can be grown in a suspension culture media. To keep cells alive and working as closely as they would in vivo, they need a lot of aid and maintenance. To avoid microbial contamination or contamination with other cell lines, cells are stored and operated within sterile conditions. Animal cell culture has become one of the most significant techniques in the life sciences, as it aids in our knowledge of cell growth and differentiation, the identification of growth factors, and the mechanisms behind the normal functioning of diverse cell types. One of the major applications of this field is in cancer research.
So, for the next point is how the animal tissue culture help in cancer research. Animal tissue culture can help in cancer research by studying the basic differences between normal cell and cancer cell, as both of the cells can be cultured in laboratory. From the basic differences between the normal cell and cancer cell that we had studied, we can figure out if the abnormal condition happened to the normal cell, the causes of the normal cell become cancer cell, and how to prevent the normal cell become cancer cell. So, what we can be found if culturing the normal cell and cancer cell in laboratory? First, we can know the mechanism and the causes of cancer by culturing the normal cell and induce it using radiation or chemical. Then, observe the normal cell. If the normal cell grew too fast and divide out of the control, it showed that the normal cell had been converted into cancer cell. There are specific radiation and chemical that can cause for specific cancer. So when we culture the normal cell in laboratory, we know what chemical and radiation that we use to induce the normal cell to convert into the cancer cell. Based from the experiment, we can know the mechanism and the causes for a particular cancer. Next point is, we can determine effective drug for selective cancer by culturing the cancer cell and induce the cancer cell with the drug. Then see whether the cancer cell can be destroyed or not. For example, we culture MDS cell, which is human breast cancer cell line, and induce the MDS cell with particular drugs. Then observe the MDS cell. If the MDS cell can be destroyed, it shows that this particular drug is effective for this cancer cell. If the MDS cell cannot be destroyed, it shows that this particular drug is not effective for this particular cancer cell. Thus, this shows that there are effective drugs for selective cancer. Lastly, not be forgotten that these animal cells extracted from their tissue or organ are cultured in aseptic laboratory with the conditions same as in vivo, and there are many steps should be taken while doing animal tissue culture.
Now, moving on, I will talk about the type of cancer cell that we choose, which is HeLa cell and also MDA cell, because these two cells we have used in our laboratory before. Okay, so now about the history of HeLa cell. HeLa cell was first found when Mrs. Henrietta lacked cervical carcinoma in the early 1950s. Normally, cancer cell would divide a few times and die off after additional studies could be done with them. But for Henrietta cell line, it just keep on dividing and dividing just so long as they were fed the right mix of nutrients for them to grow. So Henrietta cancer line have become the first human cell line to be established in culture and uh, gain named them after the two letters of her name, which is Hela. And as for MDA cell, it is established from a pleural effusion of a 51 years old Caucasian female in 1977 with a meta with a meta with a metastatic primary adenocarcinoma and is one of the most commonly used breast cancer cell line in medical research laboratory. This MDA cell is highly aggressive, invasive, and poorly differentiated triple negative breast cancer. Okay, so now I will talk about the specialty and also advantages of the cancer cell. So first we go to HeLa cell. HeLa cell is the first immortal human cell line that have been cultured before, and it also proliferate abnormally rapidly. Also can test the effects of radiation, cosmetics, toxins, and other chemical on the human cell. And it has been used in instrumental in gene mapping and studying human disease, especially cancer, because it is an immortal cell line. Okay, and finally, structure. It is an encouraged depending cell line. And finally, for the MDA cell, it is one of the human breast cancer cell line and most commonly used breast cancer line in medical research laboratory. This cancer cell lack the growth factor receptor, which is the HER2, that they represent a good model of triple negative breast cancer. And MDA cell also one of the encouraged depending cell line. Okay, that's all from me. Moving on to the next presenter.
Okay, Assalamualaikum and greetings to Doctor and my fellow classmates. My name is Hayatun Nabila binti Norjan and my metric number is 202650. So today, I'm going to present on the common research method used for the cancer cells research. The first method is RT-PCR, also known as reverse transcription PCR. In this method, biomarkers are used in the early clinical drug development, which is in the phase 1. Biomarkers plays an important role in transition of potentially discovered new drugs into clinical drug development. It enables early proof of concept studies and also better prediction of dose range in larger phase 2 and phase 3 studies. In this method, the data obtained are in form of PK, which is pharmacokinetic data, and PD, which is pharmacodynamic biomarker data that demonstrate the relationship between drug concentration and drug effects. Next is ELISA. ELISA is an immunoassay technique used to detect or quantify substance based on immunological reaction. It is a readily automated technique that is capable of high throughput quantification of analyte concentration. ELISA often perform on 96-well microplate in a standard 8-row by 12-column format. The mechanism can be observed by the color changes of solution in the wells, which is then measured by the spectrophotometer. The third method used commonly in the cancer cell research are the Western blot. This method is used in combination with other key antibody-based detection technique such as ELISA. It is to confirm the identity of target protein and also to observe the response of particular protein to drug treatment, which provide confirmation in research and diagnostic testing. Lastly is the CRISPR-Cas9. CRISPR-Cas9 is a gene editing technology which is efficient, simple, and programmable as it can edit target sequences at high rates, easy to use and design, and also for precision targeting. CRISPR is a combination of synthetic gRNA, which is guide RNA, and Cas9 enzyme to edit DNA precisely and easily. CRISPR also helps to identify target molecules for potential drugs by activating or inhibiting genes related to the disease. CRISPR could also create cellular model systems that precisely mimic to the disease, which is enable the researchers to verify the safety and efficacy of the drugs. Next, advanced research method. For the advanced research method, the method that I found is the alpha LISA. Alpha LISA is an advanced method from the conventional ELISA, in which this method helps reduce the screening cost and increase throughput during drug discovery process by eliminating the need for multiple washes. This method is also suitable for miniaturization and automation. Alpha LISA can be set up as sandwich or competitive ELISA. It is a rapid and simple quantification of analytes as it reduces hands-on and total assay time. Alpha ELISA is crucial when quantitative detection of analytes must be performed on sample available in limited quantity. That's all from my part. Thank you.
Moving on, next is the comparison of the different aspects of 2D and 3D cell culture. 2D cell culture has been the method used to culture cells since the early 19th, which plays a vital role in research but has many limitations due to 2D models inaccurately representing tissue cells in vitro. Another method known as 3D cell culture has shown improvement in studies targeted toward morphology, proliferation, differentiation, and drug metabolism. All of this made possible by 3D culture's capability to model a cell in vivo while being cultured in vitro. 3D cell culture has also been useful in cancer research. The first aspect is the cell shape. The cell in 2D is flat and elongated since the cell can only grow and expand two-dimensionally, which is also why cells grow into monolayer on the plate, whereas in 3D culture, natural shape of cell is preserved. Cell grows in 3D spheroids, it contain multi-layer. In cancer research, spheroids are used as multicellular tumor spheroids models to study solid tumor biology. Secondly, cell exposure to the medium. In 2D, all the cells will receive the same amount of nutrients and growth factor from the medium, and this causes most cells to be in the same stage of the cell cycle. Differently, in 3D culture, nutrition does not have to be equally divided amongst all cells, but can be if needed. The core cell often remain inactive since they receive less oxygen and growth factor from the medium. This process resembles the core cell in tumor cells.
making it possible to mimic the behavior and structure of a tumor cell in vivo. Next, in 2D cell culture, differentiation is poor, while in 3D culture, cells are well differentiated. As for drug sensitivity, in 2D cell culture, cells often have little resistance to drugs, and drugs are not well metabolized, whereas in 3D culture, cells often have more resistance to drug treatment. Drug metabolism is much better, which gives a more accurate representation of the drug's effects. Next is the cell proliferation. In 2D, cells proliferate at an unnaturally rapid pace, while in 3D, the proliferation rates are realistic and can be high or low depending on techniques and type of cells that being studied. From the aspect of cost, for large-scale studies, it is much cheaper to use 2D than 3D culture. 3D cell cultures are typically more expensive than 2D cell culture and require more time. However, 3D cell culturing reduces the differences between in vitro and in vivo drug screening, decreasing the likelihood of needing to use the animal models. Next is apoptosis in cell. Apoptosis is a form of programmed cell death. In 2D cell culture, drugs can easily induce apoptosis in cell, whereas in 3D, higher rate of resistance for drug-induced apoptosis. Last but not least, usage and analysis. 2D cell culture is highly replicable and easily interpretable and better for long-term cultures, while 3D cell cultures can be difficult to replicate experiments and to interpret data.
In conclusion, cancer research is research into cancer to identify causes and to develop strategies for prevention, diagnosis, treatment, and cure. By this research, scientists can determine the effective drugs for selectively destroy only cancer cells. The common research methods to identify the suitable drugs for a certain cancer cell are real-time polymerase chain reaction, ELISA, Western blot, CRISPR-Cas9, and recently, researchers found a new and advanced research method, alpha-LISA, which washing array technique is not needed. The cancer cell types that are used for cancer research are HeLa cell and MDA cell. The development of techniques for growing mammalian normal and cancerous cells in the laboratory, animal cell culture has had a great impact on cancer research. The animal cells extracted from their tissues or organs are cultured in aseptic laboratory with controlled environment conditions. It has enabled experiments aimed at understanding single cells or defined mixed cell populations to be done in the laboratory rather than in animals. Tissue culture is the ultimate pioneer of so-called alternatives to animal experiments and is now a much more fundamental part of cancer research than work with animals. For over a century, traditional two-dimensional cell culture was used in drug discovery. 2D cell culture is the conventional approach that most analysts are familiar with. It involves securing, nourishing, and growing cell cultures on a flat surface, such as the bottom of a petri dish or flask. Literally, a new dimension to cancer research has been achieved by the advent of three-dimensional cell culture techniques. This approach increased manifolds the ways in which cancer cell lines can be utilized for understanding complex cancer biology. 3D cell culture techniques are now the preferred way of using cancer cell lines to bridge the gap between the absolute in vitro and true in vivo. Now we can look forward to a period of time when there will be extraordinary advances in our ability to prevent or cure cancer.
That's all from us. Thank you.